So, so today I would like to tell you something about dynamic 3D applications in cell culture. And just for a quick start, I would like to tell you a bit about Ibidi because maybe not all of you are familiar with our company. So our products focus on the imaging of living cells, and we offer a lot of labware which is specialized for optimal cell growth and imaging like chambered cover slips, but also we have cell-based assays, for example, for chemotaxis behavior. We also offer some reagents like actin markers or immersion oils, but then another big part of our portfolio are stage-stop incubation systems for live cell imaging and a pump system for the cultivation of cells under flow. Today I would like to present you two new products from our labware portfolio which can be used for spheroid formation and imaging. So why is it important to study cells in 3D, and why do we want to design products for 3D cell culture? 3D cell culture is more physiological compared to two-dimensional cell culture, where cells grow on a flat surface. And especially for some applications, it is important to study the cell interaction in 3D. The advantage of 3D compared to two-dimensional cell culture is that the cells receive signals from all sides like it is in the body. One possibility is to image cells in the 3D cell matrix, for example, in a 3D collagen, which is very similar to the extracellular matrix in the body. This is also called scaffold-based 3D cell culture, and in this little, where's my cursor? Here. In this little movie here, we see an example of cancer cells in a collagen matrix. The cells are labeled with actin in green, and in white you can see the collagen fibers imaged with a confocal refractive microscopy. Another possibility is to cultivate the cells in aggregates or as spheroids, independent of an extracellular matrix. The standard methods to generate spheroids are the U-bottom microtubule or hanging drops. But for the imaging, the spheroids need to be transferred to a flat surface. And second, if you want to look at things which happens during spheroid formation or growth, for example, morphology changes, this is of course difficult. So therefore, our goal was to develop products that are suitable for both the generation and parallel live cell imaging of the spheroids. Next slide. So the first step for our scientists to design such products was the development of a surface which is cell repellent, meaning it doesn't allow any cell adhesion. So roughly 1.5 or 2 years ago, we developed our bioinert surface. This is a 200 nanometer thin hydrogel layer on the Ibidi polymer cover slip. It is long-term stable and doesn't allow any cell or protein adhesion. In addition, it preserves the high optical quality of the polymer cover slip and has also no autofluorescence. So we see here in these two examples on the left side, two cell types. So on the left, we have the cells grow on the Ibidi treat, and they form a 2D layer, compared to the images on the right side, where the same cell types were grown on bioinert. And here the cells form aggregates because they cannot attach to the surface. So with this bioinert surface in our hands, we were now able to develop new tools for spheroid formation. One product which we launched last month is the Spheroid perfusion slide. This slide has 21 microwells with a bioinert surface that are connected by channels. So always 7 of these microwells are connected by one channel. And the microwells have a diameter of 800 microns. And of course, the bottom is a cover slip, a polymer cover slip, which shows the imaging. So the geometry of the microwells allows the formation of spheroids just by seeding cell suspension, as you can see in this little upper movie here. Alternatively, it is also possible to transfer complete spheroids. And of course, fixing and staining is also possible, and we see cell stained spheroid. So because, as I said before, the microwells are connected by channels, it is possible to cultivate the spheroids under perfusion, or let's say under constantly exchange the medium. And as you can see in this simulation here on the right side, the flow is only in the upper part of the microwell, which we call perfusion dominated area. But there is no flow in the little niche where the spheroids are growing. So this means the spheroid is not flowed away or affected by the flow, but it's still the optimal nutrition and gas supply is ensured in this little niche here. So what are the advantages of the perfused cell culture? This I want to demonstrate in this slide. We see here spheroids from a fibroblast cell line, which were grown for 14 days with regular medium exchange once a day on the left side, compared to continuous perfusion on the right side. And what we found is that the spheroids with constant medium supply grow faster and bigger and are more compact and stable compared to the spheroid which were not under constant flow. So we think that especially for long-term cell culture, the perfusion of spheroids can make sense. Another way to generate arrays of 3D cell aggregates or spheroids is our micropatterning technology, which is available in the 8-well slide and the channel slides. So for the micropattern surfaces, the basis is as well the bioinert on a polymer cover slip, but there the bioinert surface is patterned with immobilized adhesive molecules. So this allows to restrict the cell attachment to specific areas on the surface. So for example, here are the cells patterned in the shape of our logo. But other examples of possible patterns are single cell spots, like here on the left side, or stripes, or multi-cell arrays with big round cell patches. So how can the micropattern surfaces are used for spheroid formation? So here we take the multi-cell pattern with larger pattern or patches. In this example movie, we used adhesive spots with 200 microns in diameter, and we seed the cells on top of the pattern. And then the cells assemble on the adhesive area spots and create first a monolayer of cells. And then they grow further, either by cell aggregation, like it is here in the movie, or also by cell proliferation, if, for example, less cells are seeded. And as illustrated on the scheme on the bottom, the aggregates form stable round, or the cells form stable round aggregates or spheroids over time that are attached to the surface. By using this method, the spheroids are immobilized, and this is a big advantage because it allows, so the immobilization of the spheroid allows tracking of individual cells within the spheroid. And when using a channel slide, the immobilized spheroids can also be perfused. So also in this case, the perfusion influences the speed of the spheroid growth. This is what we found. And in the top images here, the spheroids were cultivated under static condition, and in the lower panel under constant media supply. And also here, the spheroids are more compact and stable compared to the static condition. So to summarize this, we think we have now 2 new tools which allow the parallel cultivation and real-time imaging of spheroids. Both so the spheroid perfusion slide and the micropatterning slides allow long-term cultivation of cell of spheroids with perfusion. The micropatterning also immobilizes the spheroids and therefore allows to track single cells because the spheroids are not moving around. One thing I didn't mention yet is that also both slides can be used for the co-culture of spheroids from different cells or spheroids with immune cells, for example, T cells. So this brings me already to the end of my presentation. I want to thank the AROX team for the great organization of this event and for the opportunity to speak today. And thank you all for your attention. I'm happy to take your questions.
Great, thank you very much for that. A very interesting talk. So we'll see if we have some questions. I'll start off with one from myself. The structures you were talking about on the patterned cell slides, are these things which you just have fixed designs, or can people choose to have their own custom designs on these? And if so, how do you actually create the patterns on there?
Mm-hmm. So we have fixed designs that we offer, but actually all designs are possible. So you can also have customized designs. Mm-hmm. So we have a few designs like the bigger spots or for single cell arrays. I think also the stripes, we have them available just to buy, and whatever pattern you can think of, you can order actually as a customized slide.
Mm-hmm. Okay, and the comment from Philippa saying we'd be very interested in having some samples of your spheroid cells to test with our Unity confocal along with our collaborators at Imperial College London. Could you let us know how we could arrange this? Or maybe you should let her know offline. But,
yeah, you can just send me an email to either to info@ibidi.de or to tech support at ibidi.te. Then so the email will end in my mailbox, and I'm happy to send you some samples.
Yeah. And I think Mika's got a question. Could these slides be used for bacteria?
We think so. So we have many customers who are using the Ibidi treat surface and the polymer for bacteria, but also I think or you mean the micropatterning, so that they stick to the micropatterning. Sorry.
I think that's what the question's about.
Okay. So for now, we only offer the patterning with RGD. So it's optimized for cells binding to fibronectin. But it would be interesting to learn which adhesive molecule you would need for the bacteria. Mm-hmm. So then we can have a look and discuss with our R&D if it's possible.
Mm-hmm.